Hello again. This little lecture is on the topic of statins. Statins are probably one of the classes of drugs that the vast majority of people will have heard of. There's a huge amount of debate in the media and within journals and research circles as to what the appropriateness of their use is, whether they should be getting prescribed routinely, etc, etc. Now we're going to leave that debate for another day. That's not something we're going to get involved in because we're focusing primarily on the pharmacological aspects of these medicines. So we know that statins are cholesterol-lowering drugs. So why are you taking them and how do they actually do that job? And what we've got here is a dozen or so slides just to talk us through those key elements. So cholesterol. We need cholesterol. Cholesterol is often seen as the bad boy of biological science. Cholesterol is a bad thing. Too much cholesterol, it's fatal. Causing plaques, it's going to kill you. But actually we need cholesterol. It's a vital, essential component of cell membrane structure. And also, it acts as a precursor to many of the steroids that the body makes, including the steroid hormones, glucocorticoids, etc. So we need them. So that's the first thing. Cholesterol is needed, is essential. Now, the other thing is, we're also going to talk about the fatty acids. Now, fatty acids are an important energy source. And in the plasma, they are esterified with glycerol and they form triglycerides. And so therefore, when we get reduced calorie intake, we can take the triglycerides, we can break them back down and we can release the fatty acids that we need as an energy source. So the fatty acids are also equally important. Cholesterol, as the name implies, belongs to a class known as a steroid. And it's mainly synthesized in the liver, but we can also obtain it in the diet. The thing is, we don't really need to obtain it in the diet, and that's where the problems arise. We're taking too much in the diet, and that's where the high levels can occur, and those high levels are what can then start to form the plaque. So the synthesis in the liver to make the cholesterol involves several enzymatic steps. But whenever you've got a sequence of enzymatic mechanisms, there is always one that is called the rate-limiting step. That's the one that the speed of the reaction determines the overall reaction of the process. And the rate-limiting enzyme in the synthesis of cholesterol is the enzyme HMG-CoA, which is beta-hydroxy beta-methylglutaril coenzyme A reductase, or HMG-CoA reductase to give it the short form. And it's this bit here will be plenty. If you can remember all that, that's great. But the key words are HMG-CoA reductase. Now, if you get abnormalities in the lipoprotein metabolism, you can produce an excess of circulating cholesterol and circulating triglycerides. Now, the clinical importance of that is that they do have a part to play in the production of atheromas or plaques, and that becomes a problem. So if you've got an excess of it circulating, more than you need to do the jobs that we've said it does, that becomes a problem, and that can lead to the production of the plaques. And again, we've mentioned plaques before when we talked about angina and the narrowing of the blood vessels, so you can see that that has a critical impact on the control and the function and the proper working of the blood supply. Now, if you get damage to the endothelium, what can happen then? Now, what can cause that is low density lipoproteins, atherogenic factors such as smoking and high blood pressure can damage the endothelium. And that damaged endothelium will cause monocyte attachment and cholesterol to accumulate. So it's that injury, so you can see why things like smoking, hypertension, etc., too much cholesterol in the diet even, that can cause cholesterol accumulation from the low density lipoproteins. And then in the wall, once the cholesterol builds up there, it undergoes oxidation. So the cholesterol is oxidized, and that produces a lipid that is both cytotoxic and chemotactic. And what that is, the cytotoxic part causes damage to the endothelium, and the chemotactic part attracts monocytes, which are white blood cells. So it's both cytotoxic to the endothelium and chemotactic to attract monocytes. Now, the oxidized low-density lipoprotein is then taken up into the macrophage, which come from those monocytes that have been attracted. And that migrates into the arterial wall. And then the lipid-rich macrophage that you've got now, which are referred to as foam cells, they will form the fatty streaks of the atherosclerosis, or the plaque buildup. Now, those streaks release cytokines. The cells secrete more cytokines, and the cytokines enhance the low-density lipoprotein binding to the smooth muscle wall. And so what happens is, it's almost like a downward spiral after that. And the endothelial injury that's caused by that process then initiates a platelet activation. And the smooth muscle cells will proliferate over the lipid-based plaque, and you'll get a narrowing and a further narrowing of the blood vessel. Now, the patterns of the lipoproteins that are referred to as atherogenic can result from dietary factors, so the food that you eat. They can be inherited disorders, which are problems with the enzymes or the receptors involved in lipid metabolism, or they can be secondary lipid disorders, such as hyperlipidemia, which can result from diseases such as liver disease, nephrotic syndrome, hypothyroidism. So the reason these are secondary lipid disorders is the hyperlipidemia comes about as a consequence of one of the primary disorders down here. Now, the drugs to treat it, if you've got too much cholesterol, you can inhibit the enzyme that's involved in the synthesis, and that's what statins do, and that's the one we're focusing on. Or you can inhibit cholesterol absorption using specific cholesterol absorption inhibitors. You can use fibrates, nicotinic acid, etc., etc. And there's a whole section in the BNF looking at drugs to treat hyperlipidemia. So if you go to the BNF, the section numbers may have changed, that gets updated regularly, but if you go to the BNF, you go to the search engine in the BNF and you enter hyperlipidemia, then this is what will come up, and you'll get a list of them. You're going to focus on the statins for this part of the lecture. So, probably the best known HMG-CoA reductase inhibitors are atorvastatin, pravastatin, and simvastatin. So what you notice there is the vastatin part is a standard name, so you recognise the name atorvastatin, pravastatin, and simvastatin. And these are just the trade names, Lipitor, Lipostat, and Zocor. So you would recognise either of those. Now if you're looking at the BNF, you could enter either those words or these words, and you would always get them coming up to tell you what they're all about. So the mechanism of action of these statins is they inhibit the enzyme that catalyses what we termed was the rate-limiting step in the synthesis of cholesterol by the liver. And that results in a fall in cholesterol. Now, the diagram here talks you through that. So what you'll see there is, on the left-hand side, we've got the hepatocyte, and the hepatocyte will, it's where you get the conversion of acetyl-CoA into mevalonate, which then gets converted into cholesterol. Now, those are two enzymes. Enzyme there, but it's this enzyme here, the HMG-CoA reductase, that is the rate-limiting enzyme. That determines the rate at which the top thing, acetyl-CoA, gets converted to cholesterol. So, statins work there to block that enzyme and therefore reduce the synthesis of cholesterol. Statins also work on the membrane of the hepatocyte to block the entry of cholesterol into the cell. So what you've got then is, that's the blood vessel, and you've got your low density lipoprotein there. That's going to come across here and get taken up, and you'll get cholesterol there. So you see the statins are working to stop the production of cholesterol in the liver cell, and they'll stop the entry of cholesterol, to make cholesterol in the liver cell. And they will also stop the cholesterol that's still left crossing out of the hepatocyte and into the blood vessel, into the capillary, where it can do damage. I've also indicated where some of the other drugs work, like the fibrates and the nicotinic acid compounds. But what we're seeing here is, the reason that statins are so effective is, what you want to do is, you want to minimise this step here. So, that going across into here, right? And so the statins will reduce this, they'll reduce that production, so this goes down, and they'll reduce this, so that goes down again. So they work in three ways to prevent the cholesterol moving across into the capillary. So as it says, they're reducing the hepatic production of VLDL, and that there leads to a reduction in circulating triglycerides. They also give a moderate increase in HDL cholesterol. So you may refer to this as good and bad cholesterol. So you get very low density lipoprotein is reduced, and HDL, high density lipoprotein, is elevated slightly. Now, they may have some other actions that can contribute, but the actual contribution of the other effects of the statins to their lipid lowering, cholesterol lowering profile is really unknown at the present time. Now, what they can do is the function of the vascular endothelium that's been destroyed or damaged by the hypercholesterolemia can be restored. Now, that's not known whether that's a direct effect of the statin or whether it's a consequence, a secondary effect of the reduction in plasma LDL levels. They can also stabilise atherosclerotic plaques by altering smooth muscle cell proliferation and migration, so that they can stabilise the plaque. And they can also decrease plasma fibrinogen, therefore they can minimise hemostasis, and so therefore that will result in the breakdown of the clot or fibrinolysis. So that minimises the likelihood of a clot forming. They are also able to reduce the infiltration of inflammatory cells into the area, and so therefore the area around the plaque is less likely to become inflamed. The pharmacokinetics of the statin is they're well absorbed from the gut, so they can be taken orally. Now, if you take simvastatin as an example, simvastatin is a prodrug, which means that the simvastatin itself in the tablet that you take has no activity. But when it goes through first-pass metabolism in the liver, the metabolite activates the simvastatin into the active compound. But then when it's metabolized again further, it becomes inactive. So you've got the thing there is, it's inactive, metabolism activates it, and then further metabolism deactivates it. And then the atorvastatin again undergoes first-pass metabolism, and in part that's to active derivatives. So whereas with the simvastatin, the metabolite, the second lot of metabolites, so when you've activated the drug, you then deactivate it. The secondary metabolites are not active, but in the case of atorvastatin, it gets metabolized to active derivatives, and therefore the metabolism process means that you've got active metabolites which enhance the duration of action of the compound, therefore prolong the half-life. They do produce unwanted side effects. They do cause gut upset, and that can include simple things like flatulence, but it can also cause severe vomiting, abdominal pain and diarrhea. In terms of the central nervous system, they can cause dizziness and headache. They can upset liver function tests, the results from that. So you can see different levels in liver enzymes, etc. And in very rare cases, they can cause hepatitis. They can occasionally cause myalgia, or muscle problems. The mechanism of that part is relatively unknown, but it might be that there's an alteration in muscle regulatory proteins, and that's caused not so much by the cholesterol, but in sort of byproducts or intermediates of the process whereby we're making the cholesterol. So the synthetic pathway within the body that makes cholesterol, there can be some of those intermediate products might be having an impact on muscle protein to cause muscle pain, etc. But that's sort of unclear. So what we've done there in that very short sort of summary is we've looked at the statins, we've looked at what the problem is with high cholesterol, and we've identified that what the cholesterol does is, so what the statins do is, they reduce the production of cholesterol by blocking HMG-CoA reductase, but they have further secondary actions to that, which reduce the amount of cholesterol that can be found in total in the hepatocyte, and also reduce the amount that's, if you like, passed from the liver cell back across and into the blood vessel. And the reduced level of cholesterol, or the less cholesterol that gets punted from the hepatocyte to the blood vessel, the less likelihood you have of atherosclerotic plaques building up within the blood vessel. So the purpose of the statins is to reduce that damage that occurs as a result of cholesterol buildup. Thank you.